All right. طيب, we are still talking about alkylating agents, بس رح نحكي هلا عن other agents that can alkylate DNA. ورح نحكي عن النيتروزويورياز. هلا, to help you, let's say, memorize exactly what we mean by nitrosoureas, can you, let's say, think of how we can draw the structure of these drugs? يعني لما تسمعوا nitrosoureas, what functionality you are familiar with راح تكون موجودة بالstructure? urea, مش مرت عليكم هاي الفانكشناليتي؟ What is it composed of? This is the structure of urea, okay? And this is the nitroso group. So nitrosourea is simply a urea group that is covalently bound to a nitroso group, which is NO. Okay? هلا, these drugs were discovered based على إنه كان في, let's say, a compound that had a nitroso group and had some cytotoxic activity. So what they've done simply is that they, let's say, developed this lead compound, and they found out when attaching urea to this compound, it gave some enhancement بالcytotoxic activity. لكن still it wasn't enough. They found out طبعا through synthesizing many analogs of nitrosourea compounds that if you attach an ethyl or a chloroethyl group to one of the, let's say, nitrogens, or in particular the nitrogen that is attached to the nitroso group, this will give rise to drugs that can alkylate a DNA. Examples of FDA approved nitrosoureas that are used as anti-cancers اللي هو اللوماستين والكارماستين. Okay? هلا, these drugs being able to penetrate, طبعا since they are lipid soluble, being able to penetrate the blood-brain barrier, they are mostly used for treating the brain tumors because they can accumulate in the brain. They are used to treat the brain tumors. Okay, فهلا بدنا نحكي عن mechanism of action and how they generate eventually alkylating agent. طيب, هل برأيكم الالكيليشن بصير via formation of an aziridinium ion? شو رأيكم؟ مالكم يا جماعة؟ انتوا كثير أفضل يعني حتى اللي موجودين أونلاين بالمحاضرة بتتفاعلوا، ليش اليوم لأ؟ Can anyone answer me? Do you think إنه these two drugs can generate aziridinium ion? بمعنى, هل اللون pair of electrons اللي على هالنيتروجين أو حتى اللي على هالنيتروجين, do you think it can attack this carbon وبتطلع عندي الكلورين وبتكون الأزيريدينيوم أيون؟
لا لأنهم
لا.
Yes. Yes, بالضبط, you are absolutely right. The lone pair of electrons of this nitrogen is very, very weak in nucleophilic or nucleophile, simply because the lone pair of electrons are actually withdrawn via resonance by this carbonyl. Or not only by this carbonyl carbon or that carbonyl group, also you can think of this another carbonyl or another, let's say, nitroso group withdrawing electrons from this nitrogen via resonance and for sure inductively. Such a nitrogen is entirely, or you can almost say that it doesn't have any nucleophilicity, فبتوقع إنه هاي النيتروزoureas ما بتشتغل via formation of aziridinium ion. What is known about its mechanism of action is the following. It's important, by the way, يعني لنفترض إنه سواء بالكارمستين أو اللومستين إنه I will be referring to this nitrogen أو acid, to this substituent on this nitrogen as R. Okay? This is what happens. راح أرسمه على أساس إنه نمشي فيهم step by step. In the next slide I will be, let's say, presenting to you exactly الميكانيزم اللي احنا هلا رح نرسمها. What is believed to happen is that this nitrosoureas, if exposed, let's say, to any base, مثلا hydroxyl أو حتى water itself, it can obstruct that proton, breaking that bond between the hydrogen and nitrogen, which will, let's say, rearrange or form a double bond with this carbon. هلا normally احنا when we do so بنحكي انه ممكن يصير في عندي breakage of this double bond to give rise to a negatively charged carbonyl. لكن in the case of nitrosoureas in particular انه انا في عندي a nitroso group اللي بيصير انه رح تنكسر الباند ما بين الكربون والنيتروجين rather than the pi bond, simply because that breakage can be, let's say, stabilized by resonance by the nitroso group. Okay, طيب عشان أو to keep track of what is happening خليني أرقم كل الاتومز عشان نعرف exactly شو بيصير عندي. Okay, so this nitrogen now still bound to R. Okay, وبينها وبين carbon number 2 there's now a double bond, وstill carbon 2 is still bound to an oxygen. ليش رسمتها in a linear form برأيكم؟ What is the hybridization of carbon 2?
لأنهم planar.
Yes. هي more يعني such a geometry.
sp2.
Yes. We consider it linear. هلا carbon number 2 الhybridization لإلها sp2. That is why when drawing it in this structure رسمتها وكأنها trigonal planar, or it has a trigonal shape. الزوايا ما بين السيجما bonds 120. هلا when you form or when this carbon breaks the bond بينها وبين النيتروجين, forms a double bond بينها وبين nitrogen number 1, okay, بمعنى إنه هاي الكربون هلا صارت مرتبطة ب 2 atoms or have 2 attachments through 2 sigma bonds and through 2 pi bonds. Remember هاي الكربون إيش الهايبريدايزيشن اللي إلها بيكون؟
sp.
بالضبط sp. okay, being sp معناها الباند انجل ما بين التو attachments اللي مرتبطين بالكربون بينهم 180, والgeometry of such, let's say, arrangement is linear. عشان هيك أنا رسمتها in this manner. Okay. Now, this specific functionality اللي نتجت معاي is called isocyanate. والisocyanate is known, as we'll see in the next slide. طبعا it's reactive. الكربون اللي موجودة عندي being bound, let's say, to 2 heteroatoms عم بيسحبوا منها electrons inductively, meaning that, or even via resonance, meaning that its partial positive charge راح تكون عندي high. This carbon is highly reactive, which makes the isocyanate electrophilic. What they found experimentally إنه الإيسوسيانيتس أو الإيسوسيانate intermediate أو product اللي بيتكون معاي due to decomposition of النيتروزoureas leads to carbamylation of proteins واللي راح نحكي عنهم later. لكن المين mechanism is mediated via another product طلع معي, واللي هو لو أنا بدي أرسمه ببلش من وين؟ يعني we already drew this part. Okay, what about the next one? شو أرسم? Can you guide me? هاي nitrogen number 3. إيش مرتبط عليها nitrogen number 3?
N double bond N.
Yes.
O عليها minus.
بالضبط. Yes. طبعا for sure that pi bond لما تنكسر may simply abstract any proton from any base, any acid around it, such as مثلا the water. Okay, what else is connected to nitrogen number 3?
Chloroethyl.
بالضبط الكلوروإيثيل. Okay. طيب, do you know what is the name of this functionality لما تكون nitrogen double bond nitrogen? Diazine. Okay, you all know that ASA stands for having nitrogens in a structure. لما أحكي diazine because I have 2 nitrogens. ولأنه في a hydroxyl attached to one of those, let's say nitrogens, we call it a hydroxydiazine. وإذا أنا بدي أكمل الاسم بحكي chloroethyl hydroxyl diazine or hydroxydiazine. Such a compound can also decompose easily لأنه لما يكون في عندي أنا نيتروجين وإن between a double bond جدا سهل إنه تنكسر الباندز ما بين النيتروجينز will for instance الكربون to generate N2. And what is N2? What is N2? مش nitrogen gas. هلا any reaction that involves generation of a gas بيكون usually favorable reactions لأنه أنا I am generating gases بزيد الانتروبي أو بزيد العشوائية بالسيستم. ف this is what facilitates the decomposition of this compound to give rise to into gas. وال OH بتنكسر برضو الباند بين النيتروجين والهيدروكسيل بتنكسر giving rise to OH minus واللي later ممكن يصير له protonation ويعطيني الwater. And the other, let's say, decomposition byproduct اللي بيطلع معاي is this compound. Okay, بس أنا لسه ما كملت رسمه. Already على هاي الكربون أنا في عندي 2 hydrogens. Sorry, أنا زدتهم. So I have carbon number 1, carbon number 2. الباند ما بين carbon 2 والنيتروجين انكسرت. The 2 electrons of this bond are now residing ما بين 2 nitrogens that you see in here. Okay, هلا already هاي الكربون عليها still 2 hydrogens والباند بينها وبين carbon number 1. تمام؟ طيب هلا بما انه الباند او electrons of this bond are now or do belong now to the 2 nitrogens وكأنه هاي الكربون خسرت 2 electrons او خسرت هاي الالكترونز. اوكي؟ هلا صار في على هاي النيتروجين 3 bonds. طيب, do you think it now will hold any charge?
Positive charge.
Exactly, a positive charge. Carbons that are positively charged شو بنسميهم احنا؟ What do we call them? Carbons that are positively charged. Carbocations. Are they electrophilic? طيب.
Yes.
What is their hybridization? Do you remember? sp2 بالضبط. Okay. عندي 3 molecular orbitals أو آسفة 3 atomic orbitals اللي هم ال sp2 that are involved in formation of bonds with 2 hydrogens and let's say another carbon. So that's the carbocation carbon. Its hybridization is sp2 بمعنى إنه في أنا عندي 3 sp2 orbitals involved in formation with bonds with the 2 hydrogens اللي شايفينها هنا والother carbon أو carbon number 1. طيب, what else? شو في كمان other orbitals لل sp2 hybridized atoms? other than sp2 hybridized orbitals. هلا any sp2 hybridized atom بغض النظر carbon. Yes, the p orbital بالضبط. It will have 3 sp2 orbitals, 1 p orbital. وال p orbital بيكون perpendicular أو بمعنى آخر عمودي على ال sp2 orbitals. طيب bearing a positive charge هل p orbital اللي موجود على الكربون does it have 1 electron 2 electrons ولا neither؟ It's what؟
empty.
ما فيها دكتورة.
بالضبط. It's empty. It doesn't have any electrons. Okay. هلا based on that بما إنه it has an empty orbital واللي يعني بده electrons بأي طريقة. Okay. Would I consider this carbocation an electrophilic species?
Yes.
Yes. It's electrophilic و very very strong electrophile. مجرد ما يتكون it will interact with DNA. Okay. و in particular بحالة let's say النيتروزويورياز الانتراكشن اللي بيعمله mostly with an oxygen. هلا بالسلايد اللي بعده when we go over all the steps that we have explained راح احكي لكم ليش مثلا in particular بيعمل interaction مع الأكسجين. اوكي. طبعا مش هدخل بتفاصيلها لانه otherwise لازم يكون في عندنا باك جراوند عن الهارد الكتروفايلز السوفت الكتروفايلز و all that. يعني for the time being بس بدنا نحفظ انها بتعمل interaction او بيصير عندي alkylation actually على الاكسجين الكربونيل oxygen in a nucleic base. It's not the nitrogen. Mostly على الأكسجين. So again, in this slide, as you can see, any base أو حتى ال water molecules, if it will abstract طبعا in vivo, if abstracting that proton راح تنكسر eventually أو راح يتكسر عندي ال compound giving rise to isocyanate to this hydroxyl diazine. ال isocyanate is known, or what experiments have shown, it may interact with the amino group, okay, in lysines in some proteins, in particular DNA repair proteins that are involved in DNA repair during transcription or replication. Okay. هلا اللي بيصير انه هاي النيتروجين اللي موجوده عندي في اللايسين which is a nucleophilic can attack this electrophilic carbon وبتنكسر الباند بينها أو الباي باند ما بين الكربون والنيتروجين. So this is, as you can see, the bond between the nucleophilic nitrogen and lysine that is formed. This is يعني عشان أرقمهم وما to keep track. Let's say this is 1 الأكسجين 2 لنعتبر هاي nitrogen number 1 and this is nitrogen number 2. So here's the bond that is formed between nitrogen number 2, carbon 1. This is the atom 2, which is the oxygen, and this is the nitrogen 1 or the nitrogen in the isocyanate. Okay? طيب. A main mechanism via which those drugs act through is via formation of the carbocation intermediate. Decomposition of the hydroxy diazine will eventually give rise to this alkylating agent. Now, this positively charged carbon or carbocation being very, very, very reactive. يعني أنا مثلا لو بدي أقارنه مع الأزيريدينيوم ion, okay? الأزيريدينيوم ion أو الكربونز بالأزيريدينيوم ion they actually have a partial positive charge لأنهم الإلكترونز withdrawn from this positively charged nitrogen. يعني ما بيكون على هاي الكربون a full positive charge. لكن في حالة النيتروزويوريز they will give rise to carbocations that do have a full positive charge. فin terms of reactivity بيكون way more reactive. Okay? And because they are way more reactive, or in particular, we call them or we consider them hard electrophiles, راح يتفاعلوا مع hard nucleophiles such as, let's say, the oxygen in guanine. The oxygen in guanine. Remember بالمحاضرة اللي قبل حكينا إنه let's say we did identify nitrogens as being, let's say, the nucleophilic species in those nucleic bases. Only few cases can interact with the oxygen, and this is an example of it. هلا ليش الكربوكاتيون أو this carbocation carbon will alkylate الأكسجين in guanine. The reason for that يعني حتى أنا أشرحه بدي أشرح الدفرنس أو what we mean by hard electrophile, hard nucleophile. This has to do with the shape of the orbitals اللي موجودة, with the energies of the LOMOs and the HOMOs. يعني لأنه أنا بعرف إنه ما عندكم background عن هاي التوبكس أو الترمز, ف it would be almost يعني impossible إنه نفهمها بهاي الطريقة. This is actually the explanation for it, إنه ليش this carbocation will interact with the oxygen. For the time being, let's keep it, or let's just understand إنه this carbocation being highly positively charged أو completely positively charged, specifically راح يتفاعل مع الأكسجين أو الكربونيل oxygen of the amide in guanine. Okay. طيب هلا is there another electrophilic site بهالcompound other than this carbocation carbon? هل بعتبر هاي الكربون electrophilic؟ هل بعتبرها electrophilic؟ Yes, because you have a chlorine that is withdrawing electrons inductively, والchlorine is a leaving a group. Okay, being this part of this molecule being covalently bound to a guanine, فرح تكون هاي الكربون وكأنها attached to the DNA, موجودة دائما حوالين النيوكليك bases. فرح يزيد الشانس من إنه هاي الكربون يصير عليها attack من let's say النيوكليك bases اللي موجودة بالDNA, و in particular with the nitrogen number 3 بالcytosine. Okay, leading to cross-linking between DNA as if it's forming, وكأنه أنا ربطت النيوكليك bases بالDNA via an ethylene bridge. All right. طيب. Streptozotocin, which is also a nitrosourea. كيف ميزت إنه nitrosourea? Because I have a urea that is attached to NO. طبعا you have to know by default إنه this NO stands for N attached by a double bond with an oxygen. الstreptozotocin لو أنا مثلا بدي أقارنه على سبيل المثال, وخليني أرسم لكم إياه عشان تسهل المقارنة. لو أنا مثلا بدي أقارنه ب one of those nitrosoureas that we have discussed earlier, such as, for instance, اللوماستين. إيش variations in structure? By the way, streptozotocin is a natural compound. It's isolated from nature. إيش variations بالstructure بينهم? What do you think?
السايد شين مرتبطة مع الأن.
Side chain? What do you mean by the side chain?
قصدي السلسلة المرتبطة مع الأن الجهة اليمين.
Yes. Do you know what this part here resembles what? بيشبه إيش؟ It's a sugar. وبيشبه, it's actually a glucose or a glucose, let's say, substituent on the nitrogen. Okay. Now, the nitrosoureas do not have, streptozotocin doesn't have the chloroethyl. All right. طيب. What is specific about the streptozotocin? It's used for treating pancreatic cancer. برأيكم ليش؟ هل له علاقة بالجلوكوز part أو الجلوكوز, let's say, unit that is attached to streptozotocin?
I think لأن البنكرياس بفوت شجر أكتر.
بالضبط. What they found out إن الstreptozotocin have very good affinity to glucose transporters اللي موجودة عندي في البنكرياتيك cells, which means إنه راح يصير في عندي selective uptake into those cells. So that is why, let's say, it has high selectivity for treating the pancreatic cancer. Okay. طيب لو أنا بدي أطبق mechanism of action in terms of how, let's say, what is generated by the decomposition of nitrosoureas. هل بتتوقعوا بصير في عندي cross linking في حالة streptozotocin؟ هل الستريبتوزوتوسين بيعمل alkylation للدي ان اي؟
بيعمل alkylation على one strand بس ما بيعمل cross linking.
بالضبط. yes. and the specific alkylation اللي بيصير ايش؟ Do you know? هل بيعمل methylation, ethylation?
Methylation.
Methylation. Absolutely correct. So if you are to apply the steps that leads eventually to the formation of the alkylating agent, eventually راح يطلع معاي, let's say, this intermediate hydrogen double bond OH that will decompose بيعطيني ال N2 gas, ال OH minus with CH3 plus. That's the carbocation which will alkylate the DNA. Okay, وفقط راح يصير أو راح يعمل alkylation to one base pair with that's it. One, let's say, nucleic base with that's it. Or in other words, the streptozotocin is involved in only methylation of DNA. ما في عندي another active site or another electrophilic site in this side chain. Okay, such as for instance a carbon that is attached to a chlorine, فما راح يكون في عندي cross linking.
طب دكتور لو احنا احتفظنا بالإثل الكلورو إثل بالستريبتوزوتوسين يعني structure مش بيكون أحسن بالactivity بصير cross linking؟
I would expect yes. هلا الستريبتوزوتوسين as I have mentioned is a natural compound. يعني to tell you the truth أنا ما رجعت الليترشر أشوف إذا أي حدا فكر إنه to substitute الميثل with a chloroethyl. يعني it's a very very valid argument أو approach إنه كيف أنا ممكن أحسن أو خلينا نحكي تتحسن السايتوتوكسيسيتي associated with streptozotocin إنه أنا أبدل ال CH3 بكلوروإثيل. هلا in that matter if this analog حدا فكر فيه وحدا let's say عمله in that term راح يصير definitely cross linking. بس يعني صراحة أنا مش updated بالليترشر في حالة مين عمل development أو optimization للستريبتوزوتوسين. يعني this is a good thing. يعني I may look it up إذا لقيت أي شيء definitely بخبركم عنه.
Okay.
Alright. طيب ننتقل هلا to another alkylating agent واللي هو البيوسولفان. Alright. طيب where is the electrophilic part في البيوسولفان؟ هلا I will help you to understand or how you can relate the structure to the name of the compound. Where is the electron? Two carbons, yes. Which ones?
Attached to the oxygen.
Yes, exactly. So those two carbons in here are electrophilic carbons. So by the way, this compound is a symmetrical compound. يعني if you drew a line in here, you will be splitting the compound into two identical or symmetrical parts. هل تتوقع البيوسولفان بيعمل cross linking بالدي ان اي؟
يس بيعمل
100% because it has two electrophilic sites. Okay. هلا برأيكم البيو stands for what بالسولفان بالبيوسولفان؟
butyl.
بالضبط. That butyl linkage between those two groups is what bu stands for. هاي الجروب ايش بسميها؟
sulfonate.
بالضبط sulfonate. Thus the name sulfon. Okay. طيب. Why is this carbon electrophilic? Why is it electrophilic?
Because it's attached للsulfonate.
لا هي مش attached.
attached للsulfonate which is very strong electron.
بالضبط. It's a
يعني
very
مع الأكسجين.
yes, it's a very strong electron withdrawing group. Those oxygens and sulfur, and add to that, that bond is very very weak لأنه ببساطة if broken, the generated leaving group in that manner will be highly stabilized طبعا inductively and also via resonance. Being a very good leaving group, okay, this will facilitate الاتاك على هاي electrophilic carbons via nucleic or via nucleophilic nitrogens in nucleic bases بالDNA, such as for instance nitrogen number 7 بالguanine. Okay. هلا لما يصير عندي backside attack from this nucleophile على this electrophilic carbon وتطلع السلفيت at the same time. Do you remember هاي الميكانيزم احنا describe it as what? Or what is the term used to describe this mechanism? When a nucleophile attacks an electrophilic carbon and the leaving group leaves at the same time. Will attack
nucleophilic substitution.
Yes, it's definitely a substitution reaction, a nucleophilic substitution reaction. When the attack is a backside attack وبيطلع عندي leaving group at the same time, specifically we call it SN2. Okay, so the mechanism for the interaction of guanine bases in DNA with busulfan is actually or simply a substitution or SN2 mechanism. Okay. Now, the formation of this أو لما يصير في عندي let's say alkylation to nitrogen number 7 of guanine, being or having another electrophilic site can also now interact with another nitrogen 7 in another guanine, giving rise to eventually cross-linking. Okay. آخر let's say two compounds رح نحكي عنهم in terms of those compounds that alkylate DNA اللي هم الدكاربازين والبروكاربازين. ضروري برضو كمان نعرف what is the alkylating agent that is being generated. رح نحكي عن الميكانيزم that leads to it only for the case of the carbazine. Okay, البروكاربازين I will show it to you just to see أو حتى بس تشوفوا إنه eventually alkylating agent هو نفسه الانترميديت اللي بيطلع معي هو نفسه that's it. لكن الميكانيزم شوية بتختلف عشان هيك أنا ما راح أحكي عنها. اللي مطلوب منكم في الامتحان is how the alkylating agent is generated from the carbazine. Okay. طيب. أول step بدنا نعرفها أو نكون حافظينها. خليني let's say refer to this entire part as R عشان بس أسهل our description of the mechanism. أو في حالة الدكاربازين by the way the other R is also a methyl. خليني بس أعيد رسمها لأنه definitely راح نحتاج more space. The first step that happens is not spontaneous decomposition أو خلينا نحكي it rearrange itself. أول step بتصير عندي simply اللي هي أو البايو أكت أو خلينا نحكي الactivation أو road to generating the alkylating agent occurs via simply alpha hydroxylation of the carbon that is adjacent to a nitrogen. فأول step راح تصير hydroxylation of one of those methyls أو بمعنى آخر السيتوكروم أو let's say metabolizing enzymes will substitute one of the three hydrogens attached to the methyl carbon with a hydroxyl. وكلنا بنعرف إنه the carbons that have two heteroatoms attached to it through single bonds what happens is simply decomposition بحيث إنه one of the lone pair of electrons, for instance, of the oxygen will reform a double bond وراح تنكسر عندي this one. Okay, يا إما إنه negative charge will reside on the nitrogen, but in the case of the carbazine, because I have an adjacent double bond, this is what will happen. Right. Okay. طبعا besides the other product اللي بيطلع, which is formaldehyde. Okay. برجع لك. Okay. Now, the other thing that happens, also further decomposition of the compound where it will generate what is known as the diazonium or methyl diazonium let's say intermediate. Now that methyl diazonium, what will happen, it will break down eventually into CH3 plus N2 gas, and that's the alkylating agent. So again, the first step you need to memorize in order to help you memorize the entire mechanism is the formation of the alpha hydroxylation. One of the methyls on the carbazine will be hydroxylated. Now, the decomposition of this carbinolamine intermediate will eventually give rise to what is known as methyl diazonium, والmethyl diazonium بيرجع بيتكسر بيعطيني N2 gas with CH3 أو الميثيل carbocation, and that's the alkylating agent that alkylates the DNA. Okay, so let me show you the entire mechanism that we have just explained. The first step is catalyzed by liver cytochrome P450 enzyme family, hydroxylation of the one of the methyl groups, the breakage of this carbinolamine to give formaldehyde, and this intermediate that will further decompose to give rise to the methyl diazonium ion. والmethyl diazonium ion برجع بيتكسر بيعطيني methyl carbocation, and that's the alkylating agent. Okay. وضحت؟ في حالة البروكاربازين برضو كمان بصير في عندي eventually decomposition, لكن the first step تتضمن oxidation of this, let's say what looks like a hydrazine, and زي ما حكيت احنا ما راح نحكي بتفاصيل هاي الميكانيزم. What I just want you to remember إنه also البروكاربازين, و that's why we do combine them together لما نشرح عنهم. البروكاربازين gives rise also to methyl diazonium that will eventually generate, let's say, a carbocation. أو actually بحالة البروكاربazine in particular, it will form a radical, which will also considered electrophilic وبيعمل methylation للDNA. طيب, based على اللي حكيناه, does procarbazine و decarbazine cross-link DNA?
No.
No, it doesn't. Okay. طيب خليني بس احكي shortly عن البلاتينم compounds. أنا ما راح أحكي عنها أو مش راح نكملها كلها اليوم، ومش راح أكمل لآخر المحاضرة، بس على أساس إنه المحاضرة اللي بعديكم أو الشعب اللي بعديكم أو شعبة يوم الاثنين أربعة شوي متأخرين هيك let's say few slides، فعشان نمشي مع بعض، فعشان هيك أنا بس راح يعني أدخل فيها بس مش بالتفصيل الكبير. المحاضرة الجاية إن شاء الله راح نحكي عنها بشكل أكبر، بس حبيت أول أو يعني كintroduction نحكي عن platinum based drugs أو those drugs that have platinum okay in their structure. By the way, those compounds such as cisplatin, carboplatin, oxyplatin, cisplatin بالذات, they were أو it was discovered by chance. A scientist كان بده يدرس إنه كيف ممكن يأثر الإلكتريكال current على البروليفريشن أو المالتيبليكيشن of bacterial cells اللي هي الإيكولاي. فالإكسبيرمنت اللي عملها ببساطة إنه he dipped طبعا بالجروث ميديا للبكتيريا واللي كان فيها أمونيوم كلورايد. he dipped platinum electrodes. He noticed that passing of the electrical current did inhibit bacterial replication. لكن هالبكتيريال replication أو later on they found out it's not the current اللي عمل inhibition للبكتيريال growth. اللي later on what they found out إنه اللي عمل actually أو اللي صار بهاي الإكسبيرمنت إنه الأمونيوم كلورايد interacted with platinum. Okay. Platinum interacted with the ammonium chloride. It formed an adduct which is now called cisplatin. Okay, هلا cis because the two chlorides or the two nitrogens are actually cis to each other. Our similar groups are actually cis to each other. Very important نعرف إنه cis لأنه الترانس زي ما later رح نحكي لما نحكي عن structure activity relationship. الترانسبلاتين does not have cytotoxic activity أو cytotoxicity جدا 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 ضعيفة. Alright. طيب, what happens is the following. The cisplatin, when it goes through the cell membrane of cells, inside those cells بصير في عندي substitution of the two chlorides with water. هاي البروسيس احنا بنسميها aquation. Substitution of the chlorides with water molecules, giving rise to an intermediate that is positively charged. هلا that intermediate or that hydrated cisplatin form is the one that will interact with the DNA, giving rise to formation of coordination bonds مع DNA. هلا بالسلايد اللي بعده رح نشرح عنهم ونحكي عنهم. Okay. طبعا there are also other ways أو البلاتينم مش فقط بيعمل أو let's say it interacts with the DNA بالنيوكليوس وإنما كمان anything that is considered nucleophilic such as السلفرز مثلا بالجلوتاثيون أو حتى كمان في البروتينز ممكن تتفاعل مع البلاتينم أو equated form of cisplatin leading to drug activation. يعني actually the studies have shown إنه تقريبا 98% of the dose of cisplatin given is actually inactivated. The cytotoxic activity of cisplatin اللي مسؤول عنها فقط 2% of the dose. Imagine. هلا هاي process of substituting of a chloride with water molecules برضو كمان بتصير outside of the cells أو in the bloodstream. لكن due to the variations of the chlorides يعني outside of cells the concentration is way more مقارنة with the inside of the cell. That is why هاي process جدا slow أو بطيئة جدا. فحتى لو بده يصير let's say aquation this active form of cisplatin interacting مثلا with serum albumin or other kinds of biomolecules. هاي process بتكون in terms of rate, in terms of let's say yield بتكون جدا أقل في extracellular أو مثلا في bloodstream. هلا what happens is that the aquated form of cisplatin being positively charged والDNA إيش الcharge اللي موجودة عليها from the exterior part?
Negative.
Exactly, it's negative. So this positively charged active form of cisplatin being positively charged, it will be attracted to the phosphate groups that are negatively charged. Okay? وadd to that إنه برضو كمان الأمينو groups can form hydrogen bond interactions مع backbone of DNA, which will get the cisplatin or the active form of cisplatin close and interact and have favorable interactions with the DNA. Okay? راح نشوف بالسلايد اللي بعديه how the nucleic bases in DNA, okay, will substitute the OH and form a coordination bond مع البلاتينum. صح, the type of bond, as we have mentioned, is a coordination bond. It's not considered, أو بمعنى آخر الباند ما بين النيوكليك بيس والبلاتينم لا تعتبر وكأنه أنا عملت alkylation للدي ان اي، لكن لأنه such a bond, that coordination bond is part of a covalent bond. That is why إحنا بنناقش أو still بنعتبر البلاتينم السيسبلاتين أو those platinum based anti-cancers under the alkylating agents. Having two reactive sites when the other chlorine is also substituted with a hydroxyl, it will also form another bond with, for instance, guanine or even cytosine with DNA. Okay, يعني in other words, those methylating drugs, do they cross-link DNA?
Yes.
Yes, they do, as you can see. Okay, لكن ولأنه كمان those bases that formed coordination bonds with the platinum راح تكون عندي, let's say, وكأنه السبيسر ما بينهم just one atom, they will highly kink. يعني لاحظوا كيف بيكون في عندي kink بالدي ان اي structure. اوكي, لاحظوا انه السبيسز اللي هي الماينر grooves as في الميجر والminor grooves where the platinum has coordinated with the DNA increases. So this will facilitate the interaction of DNA with many, let's say, proteins that will further degrade it, break it, form complexes that will eventually prevent the binding of DNA, RNA polymerases with topoisomerases, which will eventually lead to apoptosis and cell death of those cancerous cells. خلينا هيك to look even closer على الانتراكشن اللي بيصير. What you see in here is a crystal structure of cisplatin forming a complex with the DNA. That blue dot you see in here represents the platinum. Those are the amino groups that are attached to platinum, and these are the guanine nucleic bases that have formed coordination bonds مع البلاتينum. Here's a more detailed description of what happens. This is the backbone of the DNA. لاحظوا alternating units of phosphate and deoxy ribose. We actually should be deoxy, فما لازم يكون عندنا هون OH. There is something wrong with this picture. لاحظوا this is the platinum, the two amines that are attached to it with platinum is forming a covalent bond with nitrogen number 7, either in guanine or even guanine cytosine in some cases. Okay, هلا برأيكم هل البلاتينم long enough أو big enough حتى يعمل inter-strand cross-linking؟ ولا you think it's actually an intra-strand cross-linking?
Intra-strand.
It's intra. لاحظوا those two guanines that are forming coordination bonds مع platinum are actually both of them are on the same strand. فالcross-linking resulting أو الcross-linking اللي cisplatin with cisplatin-like molecules actually they form intra-strand cross-linking. Okay. طيب. I will stop here. والمحاضرة الجاية إن شاء الله رح نبلش نحكي عن the structure activity relationship of cisplatin and cisplatin molecules. يعني رح نعرف إيش هي the structural features that will guarantee the platinum compounds how they will interact and cross-link the DNA. Any questions so far? Does anyone have any question يا جماعة؟
يعطيك العافية دكتورة.
الله يعافيكم، يعطيكم ألف عافية يا جماعة and have a good day.